So it's my pleasure to introduce today's lecture. This is the 2026 Alan S. Michaels Lectureship in Medical and Biological Engineering. This lectureship was initiated more than 30 years ago in 1995, and it has served as a forum for distinguished engineers and scientists in both academia and industry to share their views on research and development in biomedical engineering and bioengineering. Before I introduce today's speaker, I'd like to say a few words about Alan Michaels. Alan's son, Stephen, is in the audience, so he will speak up if any, if any of our notes are wrong. But it really is a pleasure to have you with us today, Stephen. Alan Michaels was a very important part of our department's and MIT's life beginning in the 1940s when he received his SB in 1994 and the MSCEP, also known as the Practice School degree, in 1947, and his PsyD in 1948, all of them in chemical engineering. Alan subsequently joined the MIT faculty and worked with Warren K., also known as Doc Lewis, and Ernst Hauser in the teaching of colloid chemistry. After a year in industry, he returned to MIT to assume the position as co-director of the Soil Stabilization Laboratory, a new joint effort between chemical engineering and civil engineering. Dr. Michaels' research interests at MIT were innovative and diverse, including colloid phenomena, soil mechanics, and polymer permeability, and its dependence upon polymer structure. He also had a particular interest in large impact and the promise of polyelectrolyte complexes and coacervates. His efforts pioneered the development of perm-selective polymeric membranes, which led to the current great interest in membrane separation processes for gaseous and liquid mixtures in the chemical process industry. In 1962, he founded Amicon Corporation, which is now a part of Millipore Sigma. I had to update my notes because it used to say Merck Millipore, but Merck Millipore is now Millipore Sigma. And they produce filtration materials for separation processes used by the biotechnology and healthcare industries, and those products are still available today. Alan left MIT in 1966 to serve as Amicon's president. Alan's many honors and awards include election as a founding fellow of the American Institute of Medical and Biological Engineering. In addition, he held several distinguished academic appointments as well as industrial and scientific advisor positions for the food, pharmaceutical, and chemical industries. He was elected to the National Academy of Engineering in 1979, and his citation was, quote, for pioneering developments in the fields of surface, colloid, and polymer chemistry, membrane science and technology, and advanced systems for drug delivery to the human body. Alan Michaels was a pioneer in the application of chemical engineering principles to bioengineering. His leadership in forging cross-disciplinary collaboration between the medical and life sciences and chemical engineering has been recognized by the creation of this lectureship, the Alan S. Michaels Distinguished Lectureship in Medical and Biological Engineering. And now I will turn to introduce this year's Michaels lecturer, Dr. Christina Smolke. Christina is CEO and co-founder of Antheia Incorporated and adjunct professor of bioengineering at Stanford University. Prior to Antheia, Christina was a professor of bioengineering and by appointment chemical engineering at Stanford, the associate chair of education in bioengineering, and an investigator at the Chan Zuckerberg BioHub. Christina pioneered a wholesale engineering approach that enables the design and reconstruction of biosynthetic pathways of unprecedented complexity in brewer's yeast. At Antheia, she has scaled this biosynthesis technology to rebuild the supply chains for some of the most complex and valuable essential medicines known to humankind. Her impact in advancing the frontiers of biotechnology has been recognized with numerous awards. These include Nature's 10, an NIH Director's Pioneer Award, and the Novozymes Award for Excellence in Biochemical Engineering. And now I'm going to go off script, and Christina has no idea what I'm going to say now. And just acknowledge that Christina has a couple of important familial connections, academic familial connections to this department as well. She and I are academic siblings, both having come from the lab of Jay Kiesling at UC Berkeley, and her academic daughter, Katie Galloway, is on her faculty, which means, and I hate it when they call me grandma, but she has a number of academic grandchildren in the audience as well. So it really is my pleasure to welcome a colleague and a friend who's made such a really important impact that she'll describe to us today on applying chemical engineering principles to practical applications and showing how that can have quite an important impact on humankind. So without further ado, please join me in extending a very, very warm welcome to our Michaels lecturer, Christina Smolke.
Thank you, Chris, for that very kind introduction. And we'll wait for the slides to come up. Do you need, oh. There we go. Okay. It's a pleasure to be here today. It's actually been quite some years since I've been able to and been back to MIT. So I've been very much enjoying my visit and the conversations that I've been able to have both with faculty, friends, and students and postdocs here. So thank you for being so welcoming. And also, it's really an honor to be asked to give this lecture to this audience. I am going to give a little bit of perspective of my journey, starting from being an academic professor and leading labs that were doing breakthrough science, as many of us in this room have all experienced, but also then the transition of leaving academia and taking that breakthrough science and really building a company and organization around it that would deliver products to industry. These are not short journeys, they're very long, and these things take time, but I'll try to touch upon and give you a sense of both some of the challenges that were encountered, how we used different principles that really I learned through chemical engineering to solve them, and hopefully develop an organization that is really transforming an industry in a very exciting way. Okay, so the problem that we're working to solve at Antheia, and that really motivated this line of research, even in my academic labs, was on around how we produce critical molecules and materials that we rely on globally. And in particular, this talk is going to focus on our pharmaceutical supply chains. And even more particularly, we're focusing here on what are categorized, and this is a formal categorization, essential medicines. So these are lists that organizations like the World Health Organization puts out, but even the US FDA has put out its own essential medicine list. So there are country-specific lists. They create these lists really to identify the drugs that are most critical for public health, that they believe have the greatest safety data around them, and that should be really available to everyone across the world. Now, many of us, well, maybe in this room we do, but I think many people, generally the public, don't think about our pharmaceutical supply chains. We basically go to the pharmacy, we expect our kids' Tylenol cold medicine to be there, and when it's not, at least I have a freak out moment of what am I going to do? I have, a 5-year-old that's crying at home with a fever. How can there not be Tylenol cold and flu? And I've experienced that even in the past several years. I think many of us experienced that most acutely during COVID, or at least this brought this area of supply chains to even the public's mind and our government's mind. Because what happened was we had an event which led to significant spikes in demand of critical medicines, but we also had events that really broke down kind of global logistics of shipping medicines, across different countries into the US. So what people experienced is whether they were going to the hospital with COVID, had to be put on ventilators, the medicines weren't there to support that, and also very basic medicines, they couldn't find basically in pharmacies. So this led a lot of people to think, how can a country like the US not have access to basic antibiotics, basic cold medicines, ADHD medicines, et cetera? So really what we're talking here is not medicines like the latest cell and gene therapy. We're talking about medicines that many of us rely on day to day. And questions arising of how can these be breaking down, certainly even in a country like the US. So this is, even now post-COVID, I think what has been, it's being tracked more, especially in the US. And what we're seeing is that these supply chains aren't recovering. So in fact, the frequency and duration of drug shortages of these essential medicines has continued post-COVID. And we're continuing to see different events, whether it is climate events, whether it's natural disasters, whether it's events like we're seeing with the conflict in Iran, right, which is closing logistical, basically transport. We are seeing impacts on these basic supply chains. So these are problems that are not going away. And it's one that, within the US at least, is now elevated and recognized very broadly, not just as a public health safety risk, but actually a national security risk. So it's actually gotten that level of attention from the US government. And really the key point of this slide is to say a lot of this is really coming about because of how these global supply chains have evolved. And in particular, how we synthesize and manufacture the ingredients that comprise these very basic and essential medicines. And for the US, one of the key points is that most of these ingredients are not manufactured in the US. We do not have the capability and infrastructure to manufacture them in the US. And we are primarily dependent on two countries, India and China, basically for the sourcing of these pharmaceutical ingredients. So this was the challenge. This was the problem. When we thought about this, again, a lot of this came back to the ways and really how we synthesize these molecules and our manufacturing production technology for synthesizing these molecules. Now, what many people didn't realize, and again, I think many people don't realize, is we think that all of this synthesis and manufacturing happens within chemical manufacturing facilities. That is true for a significant fraction of drugs, but actually about half of our pharmaceutical ingredients that are on those essential medicine lists are produced biologically. And many of them are actually produced via extraction from natural organism, whether it's medicinal plants or even animals. And it's those supply chains in particular that we see increasingly having very long durations of failure and shortage. And this was where we really focused some of our efforts in terms of the scientific development, because we saw that we could leverage this intersection of chemical engineering and biochemistry to really develop new ways of manufacturing that could address this in more efficient ways. So I just want to give you a particular example, and it's one that motivated our work, both from a science perspective, but also in terms of what we do at Antheia. So this is what the production platform for a very significant fraction in terms of volume of our essential medicines look like that we rely on globally. This is a picture of a poppy field in Tasmania. So what are the challenges here? When you're manufacturing a complex small molecule drug ingredient, and this is your primary mode of production. Manufacturing cycle time is over 100 weeks. So it's basically a 2-year cycle end to end, including harvest time, planting time, but then extraction, and all the logistics time to get that purified material to its source. The yield is extremely low. Most of the plant material, basically biomass, is being thrown away through this process. And it requires very significant amount of land, very significant amount of water, and also pesticides and herbicides to get the yield that you see here today. So it's a very inefficient process when we think about time, but also resources in terms of how we produce. That's one example, but it's certainly not the only one in terms of really critical medicines that we rely on. So we put together this slide to just highlight a couple. The one that I started with and focused on was from the poppy fields was the thebaine. There's actually about 6 different compounds, 6 different pharmaceutical ingredients that get extracted from different varieties of poppy. Thebaine is one of them. And the global demand is over 1,000 metric tons for the medicines that get produced from this crop. So it's very significant, especially when you think about that yield. Another really big one is heparin. So heparin is used as an anticoagulant, a lot of different heparin derivatives as well. This is one that is still being extracted from animals and their intestines. Extremely inefficient process, really critical medicine in terms of public health. And many other examples in terms of there's many chemotherapeutics that we extract from plants. And of course, many people are familiar with the example of artemisinin, which is one of the very early examples of leveraging metabolic engineering to produce a plant natural product. So for my own training, as I was thinking about synthesis, and I was trained, I did my PhD work with Jay Kiesling at UC Berkeley, who I'm sure many people in the room are familiar with. I trained in chemical engineering at USC. It was actually a petroleum and chemical engineering department, so very, I would say traditional chemical engineering. I did my first jobs were at leases where we pull the oil up out of the ground and actually at oil refineries. So I really was inspired by basically how we had developed a whole science and technology around organic chemistry, chemical synthesis to be able to produce most of the molecules and materials we rely on, and also really inspired by the scale at which we had deployed this technology. But I was also struck by where there are gaps that chemistry does not provide a good solution to, and began to think very specifically about how we might be able to meld biology, biochemistry, and biosynthesis with some of the tools of chemical synthesis to be able to fill those gaps in very efficient ways. And so in particular, biology, of course, is a synthesis platform. That's what biology is doing. It's making chemicals, it's making molecules, it's making materials, and it's interacting with this. And it's developed really unique catalysts and very unique chemistries that it is able to perform under benign conditions, aqueous environments, and it's able to do this at scale, right? So what I was very inspired by was, can we begin to leverage the science of biology but build a technology around it that would allow us to deploy it more effectively at scale in ways that we could actually solve some of these very big problems? So again, we come back to the challenge. I mean, we already use biology to make these medicines, and that's the whole part of the problem. Yeah, we have really long cycle times. What if we could transform that to a really efficient production host? Instead of relying on these multicellular organisms like plants and animals, which have evolved these really unique chemistries, could we take a production host that we also have a lot of experience with as humankind, but actually then engineer and modify it to be able to produce these high-value molecules? And so we focused then on the production platform of brewer's yeast. So this would give us cycle times of less than 2 weeks end to end. And be able to produce end-to-end these products from sugar as a starting point. So that was what inspired the work that my academic lab did, or at least the body of work that my academic lab did in this space. And really where we started with was this point of, how do we take brewer's yeast, a single-celled organism that is very good at making simple organic molecules like ethanol, organic acids, everything that we value in our fermented foods, but how do we now transform it to act more like a plant, in a very specialized medicinal plant? One of the challenges that I was very interested in starting my lab and from a scientific perspective is, when you start to dig into plants and their biochemistries, they've evolved really unique biochemistries, really unique enzyme classes, and they've also, they do this in part through the evolution of different tissue types. So specific types of chemistries actually occur in different tissue types across the plants. There are different biochemical environments that are present within those different tissue types to support those chemistries. So one of the challenges we identified early on is, if we really want to recapitulate plant production or, within a yeast, how do we get the yeast to act more like a plant? And how do we begin to recapitulate those biochemical environments if we want to do this efficiently? And so this, began to give us a model of, can we begin to think of the yeast cell as we do this work as more than just a bag of molecules, but really think about the internal structures within the single-celled eukaryotic cell, right? The different compartments, the different subcellular compartments that do have different biochemical environments, and could we ultimately begin to leverage that in a way where we could have specialized chemistries occurring within those different biochemical environments? And so this, again, was the inspiration for some of the work in the academic laboratory. And it concluded from the academic laboratory perspective with a paper that we published, and I'll come back and dig into some of the science, but really just to say, of course, this is like an AI-generated version of whatever cell is going to look like, right, as inspiration. And this was the illustrated version of what a very talented PhD student did in his lab for his PhD that we published in Nature in 2020. But ultimately, what we did was really try to think about the cell again in the context of the different compartments, where we localize different chemistries, and we'll come back to that through the talk with some examples. So just to give a little bit of a highlight, and this is like my 20 years in academia summarized on a single slide, but really just to say that tackling the problem that I just laid out took a lot of time. It took about 20 years of my academic career at Caltech and then Stanford University. And I'm highlighting here some of the more, I would say, notable publications that came out as sort of like key milestones along the journey. And the one in 2015 is highlighted because that will be the one we pick up on when we talk about Antheia and what Antheia has now done to commercialize in terms of its first products. But there really was sort of a stepwise journey as we went through these in addressing the key challenges that came up with being able to really set up a bottom-up platform to program biochemistry within cells to produce complex molecules. So we started the work focused on a particular class of medicinal compounds. And this was work, again, that we had published in 2015, although there were sort of stepwise papers that led to that. But really, what we demonstrated in 2015 was microbial production and biosynthesis of complex natural morphin and alkaloids, or opioids. So when we did this work, and again, as we had started this project, we identified kind of 3 key challenges. One was the number of reactions and enzymes, so to basically engineer yeast that would produce the first morphin alkaloid in the cell, we had to engineer with over 20 genes. And at the time, that was really significantly more than what anybody else in the space had done in metabolic engineering. The other one is that the enzymes, many of these enzymes are very unique to plants. You don't see them in microorganisms. They have very specific post-translational folding requirements, very specific localization requirements. So how do you get them to be functional in a microbe? And then finally, this idea of the spatial regulatory strategies that I touched upon. So at the end of the day, to create a yeast cell that produced the bane, which was sort of the first key morphin and alkaloid, we had to engineer the yeast cells with basically over 21 genes. And I think what was interesting about this work is we didn't just rely on genes from the opium poppy, although we certainly did take genes from the opium poppy. But that's the other very, I think, powerful thing about metabolic engineering biochemistry is that since we're creating from the bottom up, we can bring chemistries together that occur across different organisms to actually produce something that can be more efficient than nature. Because our goals in terms of maximizing productivity is not necessarily the natural organism's goal. So we had genes from opium poppy, Iranian poppy, California poppy, gold thread mammals like rats, as well as bacteria, Pseudomonas. So all of these were basically, and in some cases, some of these genes and enzymes were actually engineered for improved efficiency as well. But all of those were brought together into a yeast cell. And this was what was published in that 2015 paper. So again, you can see the metabolic pathway over on the right-hand side. And probably with us being in this department, we're relatively familiar looking at these. We've seen them from other labs as well. But basically, all of this, right, it's a map. You're starting with basically the input sugar. The arrows are the enzymes that are doing the catalysis. And then you have intermediates along the way. But all of this is occurring within a single cell. Okay, so under the same conditions within that cell. And this 15 years of work of my academic lab, we were able to show production of thebaine and also production of a downstream opioid hydrocodone. And what was shown was very, very small production at the bench. So basically, we were able to get, and again, you know, with these peaks, very small, so 1 microgram per liter. This is vanishingly small, in 1 ml culture tubes, right? And this was completed after 15 years of work. But at the time, and there's been a lot of, I think, very great scientific work in this space that's continued across other laboratories. At the time, this was the most complex, you know, metabolic pathway that had been reconstructed and demonstrated in a microbial host in terms of number of genes and the complexity of the enzymes. Okay, so this was really, you know, when we did this work, we felt at least we had tackled and worked through some of the challenges that occur with how do you get the cell to stably express, you know, 20 plus genes or enzymes and not die and not feel like it's being overburdened. We also were able to address many of the challenges that come with modifying the coding sequences of these plant enzymes so that they would fold and localize correctly within the plant, within the yeast cell. But one of the things this work did not tackle was that issue of kind of spatial engineering and how we think about really purposely directing enzymes to different compartments where it would actually optimize the flux through the pathway. So we decided to tackle that in the next project in the laboratory. That was based upon the figure that I showed you that we published in 2015. And we used a different pathway from the opioid pathway to tackle that problem. It is a plant natural product pathway, but instead of morphin alkaloids, it's tropane alkaloids. These are all derived from nightshade plants, and it's just it's a completely different set of enzymes, basically. So there's not an overlap in the particular intermediates that are being produced. But what you see here is basically a different way of visualizing the pathway, right? So the field and we had been very traditional in sort of drawing the pathways, as you see on the right side. As we began to think more purposely about the engineering of where localization is happening, how do we include transporters into the membranes of different compartments to really direct flux of intermediates along the cell, we felt it was actually better to draw it in the context of the cell, where we could really highlight not just the flow through the pathway, but also when enzymes were being located to different compartments, as well as transporters being incorporated into the biosynthetic pathway, not again as to catalyze reactions, but to help direct flux more efficiently to different locations in the cell. So that's what you see in this representation here. And then the other thing Prashant brought into sort of the conceptualization of these metabolic pathways was also grouping chemistries as modules. Okay, and you can see that kind of with the dashed lines that are around certain sets of reactions. What we've begun to do and what we've seen a lot of success with is actually defining modules of chemistries that we can mix and match in the cell to actually diversify what we produce very quickly. And I'll highlight some examples of how we've used that. But we layered that in as well to the approach. And so again, the next 5 years, and they get, highlighting also just the first step took 15 years. We were able to leverage everything we learned in the first 15 years, applying this to one pathway, apply that directly to the second pathway, but then also layer on these additional issues of spatial engineering and modularization of biochemistry. Okay, so now what I'm going to do is just highlight in the context of the tropane alkaloid pathway and the more recent work. And by recent, I mean this was done, again, between 2015 and 2020. Sort of some of the kind of scientific examples that came from this work. And the first set of slides really just highlight how increasingly again, this is not unique to my lab or to Antheia, but increasingly genomics and computation allow us to very much accelerate both discovery and engineering in this industry. And so one of the challenges we find, I think in almost every example we've come across of molecules that are interesting to produce is that we don't have the chemistries or the enzymes discovered to perform those particular reactions. But we are able to leverage genomics, transcriptome data, et cetera, to very quickly search and identify for new candidate genes and enzymes that can perform those chemistries. So there was a very clear example of that in the tropane pathway, where that particular step, going from hyoscyamine aldehyde to hyoscyamine, had not been discovered. So we basically took transcriptome data across different tissues in nightshade plants. We did very simple linear regression co-expression model with 2 BATE enzymes associated with it. That gave us basically 12 candidates from this assessment that we could very, very quickly test in our yeast. And it was very clear from the data that one of them was that enzyme. And so we were able to complete the pathway in that way. Now, the next challenge we had, and one that required us to really be a little bit more sophisticated in terms of organelle engineering, was this particular class of enzyme, which is a serine carboxypeptidase-like enzyme that does the acyl transferase reaction. That really does the condensation of tropine and that glucoside acid there. That enzyme, that class of enzyme very broadly, is particularly tricky to get microbes to express. There had not been an example of this class of enzyme very broadly being functionally expressed in microorganisms. We also had a problem with it when we tried to express it in yeast. And Prashant basically tried everything. He tried a whole bunch of different derivatives, modifications, tried localizing it to all different parts of the cell, could not get it to work. Finally, he really started to look at how it was being processed within the cell. And what we found is that it basically so in the plant, it would go to the vacuole, and it would perform the reaction there. It was getting kind of caught up in the Golgi and not being processed correctly so that it would actually go into the vacuole of our yeast cells. So he developed a strategy by which he basically created N-terminal fusions to this enzyme that would mask the signal peptide on the enzyme that converted it from basically being processed as a soluble protein to a transmembrane protein. And he was able to show by both fluorescence and other types of cell biology experiments that those modifications allowed it then to get processed into the vacuole of the yeast cell and anchor in the yeast cell so that it was facing into the vacuole. And that was the only circumstances in which we were able to see functional activity of that enzyme. What we also saw is that we could put different fusion proteins onto the N-terminus, and that would affect the amount of activity we saw. And for some reason, interesting reason, again, this was not sort of hypothesized, it seemed like the activity correlated with the oligomerization state of the fusion protein that was put onto the N-terminus. So now that we actually had put this enzyme, this critical enzyme, into the vacuole, what we saw that this was a bottleneck for flux through the pathway. And what we hypothesized is that those two intermediates were not getting into the vacuole efficiently. So we began to look at, could we engineer transporters into the vacuolar membrane that would help with the transport of the intermediates into that vacuole. So he basically screened a number of known transporters, plant transporters, that are plant vacuolar transporters, that work on alkaloids, not necessarily the tropane alkaloids, because those weren't known at the time. And he was able to show that there were a couple, and in particular, one in particular, but also basically the JAT1 and the MAT2, that did increase basically flux through the pathway and production of the downstream products hyoscyamine and scopolamine. With microscopy, he was able to show that those transporters were localizing correctly to the vacuole. So that experiment got him interested in terms of whether he could identify other transporters that were specific to the tropane alkaloids that would basically do a better job than just the general alkaloid ones. And so for this, the transporters that were involved in the nightshade plants for tropane alkaloids had not been discovered. But again, we had access to the transcriptome data of nightshade plants distributed across different tissue types. So we decided to go back in and see, okay, we showed that we could use linear regression analysis to pull out missing enzymes. Could we actually use computational tools to pull out missing transporters that would be specific to this pathway? Now, transporters are a bit different, right, than enzymes in terms of how you might expect their patterns of expression at an RNA level to correlate, right? Because when we use co-expression analysis for enzymes, the basic hypothesis is going to be like they will co-express in the same tissue types. They should both increase, right, to similar levels in terms of increasing the RNA transcription level. That's not necessarily the case for transporters because they're not acting as enzymes, right? They're acting as transport. So he used that sort of difference in hypothesis to test whether you could actually identify transporters from a data set like that with a standard linear co-regression analysis, or whether you could actually start to use more sophisticated kind of trained models to actually pull out more efficiently new transporters. And so what he showed in this work was that he actually tested different types of supervised AI models and showed that an artificial neural network model trained on the data set was actually able to very efficiently identify transporters for tropane alkaloids. And he contrasted that to two other types of AI models, as well as standard linear co-regression analysis. So the neural network model pulled out 3 candidates, as you can see here. And 2 of the 3 candidates were actually shown to be tropane alkaloid transporters. So very good hit rate, also very good at minimizing the number of candidates we had to test, basically, which is also important for transporters because they're not as easy to test as an enzyme, where you just look for activity. Here we were doing microscopy and other things to kind of see if they were functioning as transporters. The linear co-regression analysis, that one gave about 100 different candidates, and it missed one of the positives. So we could have searched all 100 and identified one of the 2 that pulled out with the neural network model. But again, the neural network model allowed us to search a much smaller candidate list and caught ones that the linear regression model did not. So with the 2, we were able to incorporate those into the pathway and again, reach even more increased production of our downstream products, hyoscyamine and scopolamine. So the other things that we began to look at is how can we leverage other types of computational tools to more efficiently identify enzymes. We talked about leveraging genomics and transcriptomics with linear regression models. We also had a collaboration with a lab in Europe to basically use some of their tools, Atlas and Bridge IT, which would basically take the pathway that we had set up and first do an expansion off of that core pathway to identify how you can add enzyme activities to get to interesting products. And that gave us several different areas in terms of non-natural products. And then to search existing databases of enzymes to give us candidates that we could test for those general activities. So we showed two examples here. One is in producing nortropane alkaloids, which are interesting from the perspective of derivatization of medicinal compounds. And again, here, it gave us a handful of cytochrome P450 candidates to test. Two actually had activity. And I found this one to be more interesting and not as maybe intuitive. But we also looked at basically expanding into N-oxide derivatives of tropane alkaloids using the same tools. And so again, there's like kind of less in the literature about these particular modifications. It pulled out 3 different candidates from basically different insect species for us to test that are pests basically on plants. And what we found is that one of the 3 actually worked. And it worked on both hyoscyamine and scopolamine to basically give us the N-oxide form. So again, really highlighting that the tools that we have within this space can really make our processes and workflows for identifying enzymes to test, as well as accessing new chemistries much more efficient. Other thing I want to just quickly highlight is this idea that I kind of talked about defining modules of chemistry or biochemistry within the cell. And again, this is something we at least were very excited about, and you can see kind of how we've roughly defined them with the dashed lines there. But what we're able to show in a couple different examples that we were able to mix and match the modules that we had developed, plug in new modules with slightly different sets of enzymes to get to non-natural natural products, or non-natural plant-based natural products. And so here what we did is we threw in kind of a new module 3, as well as a new acyl transferase, and that allowed us to produce cinnamyltropine, which again is not a product that accumulates in the plants, but really just shows this ability to really plug and play. Now, not just at a gene level, but at like, again, chemistry level, right, in terms of enzymes that we've grouped together. And define where those points are that you can begin to switch things in and out. Other example here is using different, again, starting materials or inputs into pathways, but leveraging the same enzymes to get to different products. So this is based upon work that we had done with a laboratory in Germany to basically identify related enzymes in a completely different plant species. And that basically gives you a different starting point in terms of that tropine moiety. But what we could show is that the nightshade plants worked on both. And so essentially, again, it's a way that we can begin to diversify the chemistries that we use in cells. And then related to the work was one about, and this gets away from the modularization of biochemistry, but really how synthetic biology, synthetic biochemistry is allowing us to have platforms that can accelerate discovery of new chemistries very quickly. And so this was in collaboration with the group in Germany. They had been working actually to identify the corresponding enzymes in the cocoa plant for these class of products. And it had been a very long-standing body of work in their field. They were using very traditional kind of plant biochemistry assessment to do the discovery. They reached out to us to see whether they could use our microbial platform to begin to screen enzymes very quickly, and some of the computational tools that we had described. So we collaborated with them on that, and we were basically able to help them fill in and discover all the remaining enzymes in this pathway in really less than a year of work. And what that also allowed us to do then, and we're just highlighting here, is that we could then begin to compare the chemistries that had evolved across these related plant species that were producing similar tropane alkaloid products, but that had very different effects. And one of the interesting things you can see is that the two plants, Tropa belladonna, the nightshade plant, and then the cocoa plant, actually evolved to develop different chemical paths to get to very similar products, right? And that's just shown here when you see the purple path and the green path. So the green path is the way that the cocoa plant basically achieves a very similar set of natural products versus the nightshade plant. And you can see that the cocoa plant had developed this interesting shunt when you look at putrescine that basically goes through spermidine, the methylspermidine, down to the N-methylputrescine as opposed to going directly through it. And that also then can sort of indicate a bit about the evolutionary history of the plants as well in terms of when they began to diverge as you look at this. From our perspective, we like this because it, again, provides us more tools for our toolbox of different enzymes we can use to test that may actually give us different efficiencies as we can reconstruct these pathways that we have found to be very important. Okay, so with that, it's sort of, and at this point, really, we had started the company that I'm CEO of. We were wrapping up this work, and sort of that was concluding the academic work, in this plant natural product space. But really, again, as we saw this in the tools that we had developed in the lab, was building out a toolbox leveraging synthetic biology that would really give us the power to program biochemistry from the bottom up within whole cells. And again, this idea of having a very broad spectrum of enzymes that we could go to mix and match chemistries, and then being very efficient about how we modularized chemistries so we could bring them together to get to different products quickly, as well as thinking about the whole cell. Not just a bag of molecules, but the different organelles, the different biochemical environments, co-substrates they offer, and how we could bring that together to like a designed system, right? Which is not just expressing enzymes. We're really thinking very intentionally about where you locate things and how flux is moving through the cell. Okay, so I thought I would spend some time talking about the next step of my journey, at least, which is moving beyond academia, starting a company, and the challenges that have come up there, and sort of at least some perspective of how we've solved them with chemical engineering and other technologies. But also, I think some of my important learnings, at least, in doing this, which might be obvious to some of you here, but they weren't obvious to me when we started. So it gets to the point, right? So we titled this sort of Beyond the Bench, right? I think there's so much really great, exciting science going on, and science that can be leveraged for technology. I think one of the challenges I've seen in our industry is really, how does cool science, first and foremost, become a scalable, robust technology that you can deploy, right, in a very predictable way. But then I think maybe what we also forget is not just how do you get to a scalable, robust technology, but how do you then actually have a solution that can transform an industry, right? And it's not inherent, right, that all those things go together. And I think sometimes we forget those steps when we're bringing things into industry and into the market. Or at least, again, it's something that I became very aware of during this journey. So when we published this work, we were faced with the question of what do we do with it? We had a sense that it could be valuable to someone. So we were filing IP through the universities during this time. And we were faced with the question of, okay, do we license the technology to an existing company? We certainly had inquiries to do so. Or do we start our own company? We made this decision, rightly or wrongly, but I actually think it was the right decision. Now, in hindsight, they were going to start a company to do this. One of the reasons why is I was very aware of the fact that this was very difficult science. Even the science part of it was very difficult. And as I said, we're at 1 microgram per liter, right? There was a lot of improvement that still had to happen, and a lot of it from a science and technology perspective. So we needed people shepherding an organization that understood the science and technology and also had a deep passion for it. So that's when we started Antheia and made the jump to do this. So we started an organization whose mission was to be able to leverage this technology to transform pharmaceutical supply chains, to be more efficient, resilient, and agile. And that was our mission. So first question here, and this is not getting into the science, but I actually think these are really important discussions to have when you're thinking about translating a technology into industry. we had to think about what was our business model going to be? How were we going to enter the market? You have this technology, you're able to produce this. There's a lot of different ways you can bring it to market. There's a lot, and again, this is a supply chain technology, so patients are not taking the ingredient that our yeast are producing. They're not drinking, a fermented product. It has to be purified, but even then, it has to actually go through additional processing, get into a formulated drug product, go into distribution and sold. So we could enter at any point that you see here. We had a lot of people telling us, just be drug formulators, right? Enter very far down. That's where the value is. That's where you make a lot of money, right? It's true. There's huge markups in drug formulation. So you got to go there. You can't do the ingredients, right? I mean, yes, you're doing the ingredients, but you should be selling the drug product. And we pushed back on that very significantly, but it was hard because it was investors. It was other people who had very strong stakes in the company. What we felt the right decision to do, and I still stand by this, is to enter as an ingredient manufacturer, right? And to first and foremost, enter earlier in the supply chain with what is called key starting materials in this industry. And then over time, step downwards into the active pharmaceutical ingredients. And again, we had a lot of pushback from our stakeholders because KSMs are the cheapest, right, kind of ingredients. And well, if we step down further, we'll gain more value. We did this for a number of reasons. One, the regulatory requirements are lower as you go further upstream in the supply chain, and the markets are more consolidated. So from an actual entering the market, it's a much easier thing for a startup to do. So then we have to think about who is the customer. Our customer are large global pharmaceutical companies. They are extremely conservative, okay? They don't want to buy technology. They don't want to take on risk. You have to recognize that if you're going to enter the market. You're not selling them technology. You have to sell them a solution to their problem, which minimizes their risk. And that's really the second point. What do they want? They don't want technology. They don't want to disrupt anything that they're doing. They want a product that is resilient, robust, but that meets all of their existing specifications because that has minimal regulatory lift for them. That's really important because it defines what our product is and what the product spec is and what we have to meet to actually sell to them. The next question, why would they switch? If they already have access to this material, why are they going to buy from you? If you're giving them a drop-in replacement, you're not changing it, you're not actually making it better for them, why would they buy from you? That was a big question that came up. Still comes up. It's all about surety of supply. This is a supply chain, basically, play. And in pharma, they do care about surety of supply. They want something that's predictable. They want something that can meet their demand. They want something like with COVID, when there's changes in demand, that they can see that response quickly. Conventional manufacturing, because it is not based on efficient chemical synthesis, cycle times are 2+ years. Our process is 10 days end to end. Okay, so that is the selling point for them. They get that, they understand that. They've experienced the fluctuations and the pain with the existing supply chains. They understand that we can solve that problem for them. And I think that's the key point here. Why are they switching? First thing, when we talk to our customers, when investors talk to our customers, it's surety of supply. They want to have stable supply. They want stable pricing. They want to know if they need this much material, 2 or 3 years from now, they can get it at whatever, at a set price. That is enough for them to actually make Antheia their major supplier, right, or a major supplier in the space. They're willing to do it for that. Capital efficiency is secondary, but it is another point for them. It means they don't have to hold as much inventory. It means they don't have to pay, the supply chains 2 years in advance to basically start production. And then the third, increasingly, is agency and control. Having production that is more distributed. It's a big topic of conversation in the US, but it's also a topic of conversation in Europe, India. The world is changing. They do not want to rely on one country where there's a lot of controls, right, and sort of things that are done to change that. And so it so happens that this has become a very significant motivating factor, not just in the US, but globally. Okay, so all of that led us to our first product. This product now is fully commercial, and it's called the Bane. You may not have heard of thebaine because it's a key starting material, but our customers use it to make about 8 different active pharmaceutical ingredient products. It's used to make Narcan, the rescue medication. It's used to make drugs like Suboxone. It's used, which is basically used for basically opioid abuse, pain for opioid abuse, so less addictive treatments. It's used for drugs like Contrav, which is used in weight loss. And it's also used as to treat acute pain, basically, right? So these are very large established markets. We're not having to build a market, and develop a market. We're entering in them, and that was the other piece of the business model that was really key. So we're not taking risk on market. Does the market exist? It's really our execution in terms of making sure we can deliver a product that can be used as a drop-in replacement. We started here because it is a large and established market. It was also a market, and it's a market where the industry has wanted a synthetic solution forever, but synthetic chemistry could not provide that solution because of the complexity of the molecule. So it was one where the industry was willing and wanted to switch over to a synthetic version. Biosynthetic is synthetic. And it was, again, one where we would be able to provide the first biosynthetic version of this molecule to the market. So it really was sort of transformative from that perspective. Okay, so what's the challenge? The challenge comes back to where we left it off in the lab, right? 15 years of academic work got us to 1 microgram per liter in 1 mil cultures. We knew that we had to get it to over 5 grams per liter, and we had to scale pretty significantly and really to 100,000 liter fermentations. Because remember, this is like 1,000 metric ton market, basically, right? So we have to produce a lot of volume. That's like a lot of zeros, basically, in terms of fold improvement. And it's very challenging, right? So that is where we had to go, and that was the challenge. How do you build an organization that takes this challenge? And I think this is an important piece of it here, because you realize the science is key in enabling all of this, but it becomes a much smaller part of the broader organization that is needed to be built to deliver this to the industry, right? So you come back again, 15 years of research, that's like, a very small fraction of the biology that we've had to put in place to basically make this a commercial product now, right? So there's a lot of, and then you add on all the other different functions. So the biology to get to the strain, you now have process development, upstream and downstream, all great chemical engineering areas, let me tell you. Operations, now you're gonna actually manufacture. You have to think about supply chain, and the stability there. Quality and regulatory is huge. And then commercial, sales partnership. You have analytics, data supporting all of this, and underlying, it supports every single one of those operations. And then data science and automation supports that, right? So this is a very significant organization that has to be built out. I put the pharma customers there because we felt it was important to start with them very early, not to bring them the product efforts being made and say, hey, you want to buy this? We have some thebaine. But actually start years before we were even producing it to say, if we could do this, like educate them, get them kind of understanding this is real, make sure they understand we're not asking you to buy a technology, but what would the product need to look like for you to buy it? What would the price need to be? And they're very forthcoming with that information, basically, right? But that way, that information goes back to the R&D team. So it's not R&D telling commercial what they should sell, it's commercial telling R&D, this is what you need to build, basically. And the reason I would just say this, you have arrows going both ways, is that it's not like a handoff, sequential, right? Like, oh, the strain's done, now we give it to fermentation. Okay, the fermentation process is done, we give it to downstream processing to figure out how to purify it. There's cycles and iterations across the whole thing. Because commercial will come back and say, you can't have this impurity in the product, it has to go away. Downstream process will then work on that and say, okay, we need a different cell. Like, you have to modify the cell to remove that impurity so that strain engineering gets on it again. So it's like a never-ending cycle of like iteration until you finally lock that to deliver the product. But I will say that I think it's very important that you get these pieces engaged as early as possible, because they're the ones that are informing what R&D should be doing and where they should be focusing their efforts. Okay, and then finally for us, the question then is like, how do you manufacture? and this is a big puzzle. I'm going to skip over all the challenges and just say that, there's decisions, right? Are you working with manufacturing partners and contracting capacity? Are you actually investing in your own facility? You see a lot of different models in the space. We've taken one where we start with contract manufacturing because it allows us not to have to invest early on. But as we grow our volumes, then we look to make that investment so that we can basically be even more efficient. But there ultimately then what gets transferred to your partner is going to be your production strains and your SOPs. And the team, again, is very involved in this. Our team is on the manufacturing floor with our partners, making sure that everything transfers correctly and everything is being produced correctly. Okay. All of this, we started in about basically end of 2015, early 2016. Towards the end of 2024 is when we delivered the first commercial product to customers. I don't mean samples, I mean metric tons of material they were paying for, okay? And that took basically 8-9 years. So it was not a light journey to get to the commercial stage. Now though, because we've been thoughtful in terms of how we've set this up, we've set up the infrastructure so that we can now launch products each year, right? So we have a pipeline of products that's progressed. We have developed our processes so that they can run on the same lines, basically with the same manufacturers. And so we're launching our second product now, and we'll launch 2 additional products next year, and then similar cycles sort of going forward. So it takes a lot to get that core infrastructure in place. You need to hope that you can navigate the stakeholders and the patients, right, to be able to do that and show those proof points along the way. But then, basically you can continue to scale with that infrastructure. And then just a few things to touch upon in terms of how, from a chemical process engineering perspective, if we're thinking about how we leverage and use our expertise. I think some of the talked a lot about probably where a lot of us, focus our training on, and certainly I did as well, in terms of the molecular engineering of the cells. I don't want to underappreciate though process engineering and all of this, and folks basically with process engineering skills, how critical a role they play. So we took an approach, because we were innovating on the cell engineering side, that we wanted to be able to slot into existing manufacturing equipment. We didn't want to have to innovate both manufacturing, equipment, processing capabilities, and kind of the cell engineering. So we needed to make sure that we were working with our manufacturing partners to understand the nature of their equipment. What's the temperature range of their fermenters, right? How much does it vary? What's the dissolved oxygen, right, that it can maintain, these sorts of things, and ensure that we're building scaled down models in the lab that recapitulate all of that so that scaling can be successful. So we run these as fed batch processes, end to end, meaning working cell bank to kind of the end of the fermentation is about a week. Major input is sugar, but we can run, and this should be pretty straightforward, right? We just swap different yeast into the fermenter, and we can basically make different pharmaceutical ingredients within that same infrastructure. Downstream processing has been a different challenge in the industry, and certainly we've seen it as a different challenge as well. It is one that really does become more tailored for a product, but what we really focus on is ensuring that we're leveraging economical and very multi-purpose unit operations. So things like filtration, precipitation, solvent extraction, and crystallization, right? These are like the basic core things that we use, and then we ensure that we can kind of mix those together. But again, the key challenge here is that whatever final product comes out of that process, it needs to meet the spec for the customers. The good thing is, because pharma is a spec-based industry, all the customers follow the same spec, right? So you're not doing multiple products, multiple specs per customers, but they tend to be very rigorous specifications. Okay. Then I think one of the things Antheia has done very well that I've been proud of is scaling. I've seen a lot of challenges in scaling in the industry where you take it to the manufacturing facility, things don't perform as you expect, sales are not robust, titers drop by 50% or even 90%. We were able to see a really successful tech transfer and scale up, and we've done this multiple times now. So this is just data that's overlaying basically the pilot runs, as well as the runs that were done at full manufacturing scale, 120,000 liters. The data actually would look exactly the same with the 1 liter that we did internally within Antheia. But we were able to scale directly from our bench scale 1 liters to pilot of 1,000 to then commercial 120,000 and not see any hit in the performance of the system, which we're really excited about. Basically then bring this to the industry, right? Again, why did this happen, or how did this happen? It happened because of the organization that was built that I showed, where there was constant feedback and iteration across the different teams, and also where we really ensured that our scale-down models were very robust, and we did a lot of robustness testing, looking what happens across different temperature gradients, et cetera, so that we ensured what we were taking to the facility was a very robust process and could withstand changes in temperature, pH, et cetera, that you might expect. Now, this has been a very exciting launch for us. We've seen really strong demand from the industry, and our customers have been able to take our material and actually get them fully approved all the way to patients in very short time. So because of the strategy that we took, they only have to amend their existing filings. And in each case, it's been treated as a minor change to existing filings with basically no comments from the regulatory agencies. So, and this is for multiple products now. They've gotten those approvals in less than 30 days, right? But again, it was because we engaged with them so early, and we had our quality teams engaging very closely in terms of both what we gave them, but also the data packages. Okay, so as I said, Antheia is now commercial. We've been producing our first product, the Bane, for about 1.5 years now. We're actually moving to a much larger facility, a dedicated facility for our products in Europe. But we've seen very robust operations. I think it's notable for the industry, and when I say industry here, I'm talking about the industry that is making drug products from our process, not the synbio industry, because this is the first critical pharmaceutical ingredient that for them that is commercially made via a synthetic process like this, and the first time that the Bane was made available via a synthetic process. We're now launching multiple products, and we're just expanding the capacity in the next 1 to 2 years to be able to support that, and also looking to expand geographical footprint. I'll do this very quickly, but again, say when we think about the value to the industry, it really starts with that surety of supply, which is this point here, and being able to have distributed production. Production of this in Europe and US is really huge, even in India, because again, it just reforms and reshapes the existing global supply chains. Yield increase, cost increase, of course, that's important, but it really starts with that, again, the value the customers see. Final thing I'll just end with is, again, if I come back to sort of what inspired the work from a scientific perspective, it was really coming back to synthesis and how do we add another tool in synthesis that can really drive transformations of industries, right? And in particular, if we look at pharma evolution, across each century, there's been a manufacturing innovation that has really led to transformations in supply chains, first and foremost, for critical medicines. So 19th century, that was synthetic chemistry. First products to come out of that were synthetic aspirin, right? Prior to that being extracted from trees. That really launched Bayer and other companies, right? And today you now see, of course, that's a big powerhouse in drug discovery, or drug development, I should say. 20th century was recombinant DNA technology. First products there, synthetic insulin, right? Synthetic EPO. Again, it was a supply chain really play there, right? But really has led to an entire new industry of biologics within the space. And we really believe that the ability to do bottom-up programming in chemistry and cells is this 21st century sort of next manufacturing innovation. I think it has tremendous disruptive and value creation potential for a very large fraction of the drugs that we rely on. And so we're excited to pursue that. Thank you.
I have a hand over there. We'll do 2 questions.
Thank you. That was a really excellent talk. I actually read your 2015 paper for a metabolic engineering class in undergrad because it was like cited as a classic paper in the field. So it's really cool to get to hear this talk. many years later. So I guess my question is, one of the things that my petroleum engineering facing professors often said for biological manufacturing was that it just does not scale. And you kind of talked a little bit about how it does. I think one of the things that I think a professor said to me was like, oh, if you wanted to manufacture this amount of it, you would need like, 10 acres full of fermenters to get a particular concentration. I don't remember what it was, but it seemed like an astounding amount. I don't know if you mentioned, but I might have missed it, how much like substance are you now able to get with your improved process, going from the like 1 microgram per liter?
So let me give you, I won't answer that directly, but let me just give you some benchmark metrics, right? I mean, you could look at sort of the metrics I gave you of like 5 to 10 grams per liter and 100,000 liter fermenter, and you could kind of back out with some recoveries like what the yield is going to be. But it's an interesting question. I think what we show here is that it does scale. Okay, 100,000 liter, 200,000 liter fermenters, those are real volumes, right? It's not a 1 million liter fermenter, but it scales to where we need it to scale for this market. We're not producing a biofuel, right? We're not producing something where the quantity that's needed to be made is like 100,000 metric tons. Thebaine and those class of molecules, which is probably one of the larger pharmaceutical markets, is about 1,000 metric tons, right? Heparin's also pretty large, but it's in that range. Again, you're not going to get to like 100,000 metric tons. So that is something you can tackle very efficiently with this. So again, okay, if we have a facility dedicated to us in Europe that has about 600,000 liters to 1 million liters, we can be producing like 600,000. Yeah, we could be producing 300 to 400 metric tons there. That's very significant in terms of global footprint, right? And we will have multiple facilities. So 100% it scales in this space. Now, again, if we're talking about huge commodity chemical markets, I think you've got a question there. You have to get to much higher productivities than what I showed here, right? It's not going to be tens of grams. It's going to be hundreds of grams per liter. And there, I think you have more stresses on the cell. You get closer to like kind of the like optimum efficiency, et cetera. So one, I think we have to choose our problems correctly, right? And the other thing to say is that synthetic chemistry doesn't scale for these problems either, right? We're not choosing problems, or I don't think it's the best thing to choose, especially at this stage in the evolution of the technology, to choose problems where you have efficient synthetic chemistry processes, right? We're choosing problems where synthetic chemistry actually cannot provide a solution. And because of the complexity of chemistry and the number of steps, and thus where biosynthesis can have an advantage. Does that, okay.
Fantastic talk, and tremendous congratulations on what you've been able to achieve. That's really awesome. So you might expect my question with respect to what is the impact of like the synthetic biology regulation aspects in terms of the metabolic engineering, the 21 genes, tremendous number of genes, the stoichiometry matter, gene expression levels matter, stability, timing of the gene expression, resource sharing. You mentioned localization, but all of these other things that we often speak about in synthetic biology, how much impact do they make beyond, after you identify the enzymes themselves?
Yeah, it's a great question. So yes, it does matter. And actually, I can just, in the production strains, we're much higher than 21. We're probably closer to 40 or 50. Okay, so because, again, we've had to get that 15-0 improvement in the overall process. But yes, it does matter, right? You cannot, and so to get and our cells do not have a significant hit in terms of their doubling time. But that's because we're not cranking out expression, right? Because if we were, right, if we start to try to just complete, right? So yes, expression matters. And then the great thing is that we're dealing with enzymes. So we actually, in most cases, don't need really high expression, right? Because they're catalytic. So it's a sort of a different problem than maybe non-metabolic engineering, but we do think overall about, again, how flux is being driven through the pathway, where there are bottlenecks. Bottlenecks will focus on, expression, but also activity of the protein, et cetera. And it does matter because you cannot overburden the cell. But again, with enzymes, we don't have to actually produce that much. The only other thing one other thing I would just highlight is that one of the things that we don't necessarily get in the academic lab, but that is very much relevant to industry, is that process engineering actually can do a lot in terms of timing, right? Because how you seed the tank, how you feed the cells, you might not need an inducible promoter because you're doing different things with the nutrients you're feeding the cells, and that will actually start expression at some point or start production at some point, right? So I would just say from an industrial perspective, when we began to look at this, it's really the melding of process engineering and the controls that we put into the overall environment, whether it's the nutrients that get fed at what point in the fermentation, all that stuff, with the actual kind of genetic circuit part of it. And you optimize both of those and bring them together.
So we are out of time for questions. I want to thank Christina again for coming and sharing an experience that really did answer this question: how do you go from science to a scalable technology, and how do you go from technology to a solution that's going to have impact? And so we know that amongst our students especially, they are interested in those questions, and so your story certainly has given them a blueprint to keep in mind when they take their own journeys. So with that, I would like to officially honor you with this memento, recognizing and thanking you for delivering a very inspiring and informative talk.
Thank you.